Autotrophism. How to the food material to prepare the sun energy, trap the sun energy, that sun energy converted into the chemical energy. That is called solar energy converted into the chemical energy is called the photosynthesis. This is the electromagnetic radiation. The solar energy is called the another name electromagnetic radiation converted into the chemical energy. This is total process to the photosynthesis. These are always surrounded whole world, the microbes, including in this process, microbes, insects, utilizing directly or indirectly, and the final is the two producers. These organisms naturally resources harming the plants, the plant biological system resist the explanation at least all levels are all means the total development diffusion mechanism in plants. That's where the resistance against harmful act or sometimes useful acts also present in the total plant system. The plant's resistance against parasitic pathogen is the habitat. This is the pathogen and host, how to interact is that one. In the yesterday class, I'll already explanation complete the host pathogen interaction. One is totally completed. This second, what is the second interaction is there? In that one, pathogen and host response. In that both are the how to respond the disease time. Pathogenes and host. Here most of the host parasite relationship, most of the host the parasite relation reveals that the pattern of pathogenesis. These plants their part the exhibit defense mechanism just like structural and chemical process is called the defense mechanism. This is the challenged by the pathogen. Okay, అంటే how to pathogen host interaction in between, how to accept that one. These are the challenge for the pathogenist. The moment pathogen propagules come in contact with the host surface, the inherent characteristic themselves using survival natural occurring physically chemically barriers also penetrations resistance. If all penetration occurs, the host react by the different types. These different types resulting in the formation of physical and chemical barriers. Okay, some physical barriers are there, some of the chemical barriers are there. That's where the plant's defense mechanism can be studied into according to the two different types of sequences. That is disease attack them. One is a pre-existing or pre-infective defense mechanism. The second one, post-infective or induced defense mechanism. First one, this is the second one. First one, pre-existing or pre-infective defense mechanism can be divided into other two parts. One is a physical or structural barriers. The second thing is biochemical barriers. This is the pre-existing pre-infective defense mechanism. The second one, post-infective or induced defense mechanism. This is a physical structural barrier. This also can be divided into three parts. One is the physical or structural barriers. The second one, biochemical barriers. Third one is the single induced responses. See, this is the picture you can observe. This is a defense mechanism, pre-infective or post-infective mechanism. Pre-infective post between the pathogen and the host reactions. This mechanism between the pathogen and host reaction. This is a defense mechanism. In this pathogen, you can observe this one, pre-penetration and penetration. This one is the invasion, colonization, diseases, symptoms. Host, what is that mechanism is there? natural occurring, pre-existing defense structure, biochemical defenses is in the pathogen you can observe pre-penetration or penetration mechanisms. The host post-infectional structure defense, biochemical defenses is called the invasion, colonization or disease symptoms. This one, the total this extra to you can observe defense mechanism or pre-infective or post-infective mechanism. See, now, pre-infective or pre-existing defense mechanism, what are the barriers are there? How to internal functions are importance are there? I'll explain. First one, pre-existing or pre-infective defense mechanism, the physical barriers, physical or structural barriers. The first line of defense in plant, is a present epidermal surface. First one, defense plants, what happened? Epiderm, in case if you take the leaf part, in that leaf part or stem, root, anyone, the effect from the starting from epidermal surface. Outer epidermal meaning outer layer surface is called the epidermal layer. These epidermal layer, some of plants outer side cuticle layer or suberin layers also present. These surface, the epidermal surface, several characteristic of the plant surface infection as a barrier to penetration with pathogen must breach to enter the host. Okay, the pathogen enter the plant host. Now what happened? The several characters are there in this one. The plant surface function as barrier to penetrate. Now the pathogen enter into the host. Penetration with pathogen must be breach to enter the host. Now what happened? The pathogen now enter the host. What is the first barrier? Epidermis surface. The pathogen first occupy the epidermis surface sometimes to enter into the host. The pathogen enter the plant host by penetrating the epidermis along with cuticle also. In the epidermis, some plants along with the cuticle layer also. These cuticle to made up of vaccines, number of natural openings are there. These cuticle always to made up of waxes or cuticle layer always waxes to made up of waxes and number of natural openings. Sometimes in the upper epidermis openings as tomatoes are there and sometimes normal openings are the openings existing before the onset of pathogens can absorb penetration. If the pathogen succeed in the penetration, it encounter pre-existing internal structure of the barriers. The external and internal structure of barrier existing before pathogen. Okay, the external and internal structures both are barriers existing before the pathogen attack, also called pre-existing defense structure or passive or static or anti-infection structures. This is called the physical structure of the barriers. In the barrier, after epidermis layer, outer side waxy layers are there. Cuticle, waxy layer. These are to made up of the secondary metabolites. In cell wall to produce these secondary metabolites. The cuticle covers the epidermal cells of the plants and consists of a pectin layer, cutinized layer, vaccine layer. These are the layers are most important. The upper epidermis, what type of layers you can observe? pectin layers are present. Some plants cutin layer, that is the cutinized layer, and some plants made up of the vaccines also, wax layers. Cutin is composed of the fatty acids. How to cutin is composed? Fatty acids are the cutin layer are the composed of the fatty acids. Next one, waxes are maximum of long chain aliphatic compounds. Vaccine cutin also fatty acid. Waxes, this is a alphanetic compounds, very long chain alphatic compounds are present in this one. What is the use of these vaccines and cutin? The prevent the reaction of water on plant surface. What are the this one? Prevent reactions or prevent retention of the water. These prevent retention of water on plant surface, essential for spore germination time. A negative charge usually develop on the leaf surfaces due to fatty acid. This condition repels or airborne spore. This condition repels for the what type of spores? Airborne spores, propagules, that these are things. Only few pathogens are known to dissolve cutin enzymatically. Only few pathogen, not for the all pathogen. Just 20 to 30% of the pathogens are known to dissolve the cutin enzymatically. Example. Okay, what is the which one pathogens dissolve the cutikin? For example, hmm, Monilinia fruticola. Monilinia fruticola penetrate cuticle of cherry leaves but not of ginkgo biloba leaves. Ginkgo biloba meaning gymnospermic leaf. Monilia fruticola penetrate cuticle of only for the cherry leaves. You know, cherry fruit in that cherry leaves to penetrate the this type of fungi. What is the fungi? Moniliae fruticola presenters cuticle of the cherry leaves but not of ginkgo biloba leaves. Ginkgo biloba meaning this is the gymnospermic plant. The later contain abundant cutin than the former. Okay, these Fruticola solani, Fruticora pisci induced the enzyme cutinase production by specific antibiotics and inhibitor. Okay, what is the epidermal layer? What are the hydatodes? These are how to penetrate the pathogen into the host cell. Epidermal is the first layer of living host cell. Where the epidermals are there, almost if you take any part of the plant, the outermost layer is called epidermis layer. This first layer living host cell that comes in contact with the attacking microbes. The together of epidermis is due to the polymers of celluloses, hemicelluloses, lignins, mineral substance, polymerization, organization compound, suberins, etc. What are these are? These are to produce the cell wall, that two secondary metabolites. For example, potato tuber resistant to the these potato tuber causing the disease when type of but these potato diseases, tuber diseases resistant to the Phythium dihydrohydranium contain higher fiber. These potato tuber resistant is called Pythum Pythum dibarium contain higher fibers. Next one, silicon accumulation in Silicon accumulation in epidermal walls provide resistance against fungal attack. This is the suberization of epidermis. This here suberin is present. This one is called suberization. This one is called the suberization of the epidermis confirms protect in the citrus plants. That too citrus plant is the Xanthomonas citri, Xanthomonas exopodis. Both are the citrus plants. A functional defense mechanism has been observed in the various stomata open last well, various stomata open the late in the day only when moisture on the surface, leaf surface, as dried and infective propagule have become a non-functional. This is the epidermal layer. The second one is the hydatodes. This is called the another second thing is hydatodes. Hydatodes are the natural openings. These are the just like stomata like structures, but not the shape of stomata. These are the in the plants you can observe natural opening systems on the edges of leaves that serve to excrete excess of water from the interior. In the inside of the plant body, excess of water is there. In that water, they are essay entry point of the bacterial pathogen. Excess of water to removal these hydatodes. But in these hydatodes, enter into the point with the bacterial pathogen. The source of bacterial to enter in this way. What is the way? What is the pathway? That pathway is called the enter into the pathogen. This is the leaf server extremely excess of water interior. They are enter into the bacterial pathogen similar to hydatodes or the nectar hoards. Nectathodes in influence of many plants. They create sugars. In hydathode, to create the sugars, nectars, and nectars, sugars, nectars, and is serve as a barrier. Those organisms that cannot tolerate this condition and thus cannot enter through nectarious. Leaf aids on leaves and nectaries also resistance entry of pathogen. Highly various high hairlines of leaves and pods in chickpea, resistant character against Ascophyta abe. This is the groundnut varieties showing resistance to Cercospora flora leaf. Cercospora leaf spot of disease, black crust disease is there. This Cercospora leaf spot have thick epidermis, cuticle, and compact palisade layers. Palisade, palisade layers, few and smaller few and smaller stomata and higher frequency of the trichomes also adaxial surface of leaf. This one is called the second thing is hydathode. Coming to the last one, lenticels. These are so opening in outer crocky walls where the lenticels are present. These are the openings in the outer crockery walls of the plants involved in gaseous involved in a gaseous exchange. They are weak in defenses unless the croc cell within the unless the croc cell with uh, unless croc cells within them are suberized. After suberization, periderm formation, lenticels are more resistant in the invasion of the pathogen. See, these are the non-physical or structural barriers. Physical, structural or pre-existing or pre-infective defense mechanism. In that too, physical and structural barrier, wax and cuticle, epidermal layers, hydatodes, and lenticels. What are the biochemical barrier? Okay. In the physical, in the naturally occurs to the in the plants. But what is the biochemical barriers? In the plant create different chemicals which directly interfere with activities of pathogen and before and reduce the effect of pathogenesis. Therefore, by preventing or reducing infection, preventing and these chemical, and you can give the chemicals. What happened? The prevention and reducing the 1 second. These are the activities of this pathogen, reduce and effective the pathogenesis, thereby preventing and reducing infection. When give the chemicals or herbicides or chemicals as well, what happen? Reducing the infection. These chemicals and the biochemical conditions that develop may act either directly or indirectly. In case it can act the directly through toxins or lytic enzymes on the invader. In this is a directly method. Direct method, the biochemical when the stop the infection, when the control the infection, the chemical biochemical condition that develop may act either direct condition, indirect. Direct condition through toxins or lytic enzymes on invader or indirect, that is a direct method. Indirect through stimulating antagonistic plant surface microflora, such protective compound against pathogens pre-existing in plants, that is called constitutive antibiotic. What is that name? Constitutive antibiotic. That name is called constitutive antibiotics, and those which are formed in response for the wounds are called, that wound is the antibiotic. That name is called antibiotic. Where? Next one, what is the antimicrobial compound? Release of antimicrobial plants in the normal phase of growth and development release certain gaseous as well as organic substance. Plants in the normal phase of the growth and development release certain gases as well as organic substance from the epidermal surface of the leaves and roots, commonly known as leaf and root exudates. These exudates are maximum of many substances contain sugars, amino acid, organic substance, enzymes, glycosides, etc. These materials have profound effect on the nature of surrounding environment, particularly the microflora and fauna of phylosphere and rhizosphere. Although of these are substance, nutrients of microbe help in the germination time, growth several sporophytic and parasites. There are a large number of inhibitors. These are the large number of inhibitor substance also present in these exudated. These inhibitor substance may directly affect the microorganism or they may also encourage certain groups to dominate the environment and function antagonistic of the pathogen. Next one, inhibitors how to present in the plant cell. In many host parasitic interaction, pre-existing toxic substance in the host cell from the basis of resistance. In the resistance variety, these substances are formed in the abundance, while in the suspectably varieties they may be less or they may be sometimes absent also. Several phenolic components, chemical substance in the phenolic compounds, tannins, fatty acids, dienes, which already pre-exist are high concentration in the host cell, have been implicated for the resistance of young, young tissues against parasitic fungi such as botrytis. Many such compounds are potent inhibitor by the hydrolytic enzymes also. Several other kinds preformed compounds such as the saponin. Saponins meaning glycosylated steroidal. All saponin is the glycosylated steroidal or terpenoid compounds. Tomatin and in tomatin. Tomatin in tomato and avanacin in oats. Avanacin in oats. Next one, avanacin in oats. Mm, Avinacin in oats have antifungal membranolytic activity. These are the antifungal membranol activity is the present in this one. The fungal pathogen. Here many pathogens are there, but the fungal mm, pathogens that is called the fungal pathogens. These fungal pathogens in important, one second, I'll explain. The fungal, these are the tomatin in tomato, avena in oats. These are the antifungal membranolytic activities. The fungal pathogen, lack of enzymes are there. What is the lack of enzyme? Saponinase is the lack of enzyme in the fungal membrane. That one is breakdown into the saponins. These are prevent from the infecting of the host. Several preformed plant protein have been reported to act as the inhibitors of pathogen proteinosis or hydrolytic enzyme. Similarly, lectins. Lectins meaning protein that bind to certain sugars. Proteins that bind to certain sugars, causing lysis and growth inhibition of many fungi. Plant surface cells also contain variable amount of hydrolytic enzyme such as gluconase, chitinase, etc., which make causing breakdown of the pathogen cell wall components. Another one, absence of recognition factor in plants, absence of host receptor and site of toxins. First one, absence of recognition factor in plants. This is the first step infection. These first step infection, cell to cell communication between the host and the pathogen. See, observe this one. Here, communication, this is the first step, but the first communication within the process, cell to cell and between the host cell to pathogen cell. These are the many of plants of many species or varieties may not be infected by the pathogen in their surface cell, lack specific recognition factors. If the pathogen does not recognize the plant as one of its host, it may not adhere to the host surface, or it may not produce the infection substance such as enzymes that are the Aprisoria Hastorias. These are the substance that produce the host. What is that one? That is the pathogen are host Apresorius or Hastorius. These recognition molecule can be variety of the oleosaccharides, polysaccharides, glycoproteins are present. This is a host absent recognition factors in plant. Next one, absence of the host receptor site of toxin, host receptor site of toxin. In many host parasite interaction, the pathogen produces host specific toxin, which are responsible for symptoms and disease development, The molecules of toxins are supposed to attach to a specific sensitive site or receptors in the cell wall. Only the plant that have such sensitive site become diseased. Next one, essential nutrients and growth factors. The fact that many facultative saprophyte, infacultative saprophyte, parasitic saprophyte, like this. But here many facultative saprophyte and most of the obligative parasites are host specific and sometimes are so specialized that they can grow and reproduce only on certain varieties of only those plant species. Suggest that for these pathogens essential nutrient and growth factors are available only in these hosts. Absence of these nutrient and stimulus autonomically make the other varieties and species unsuitable or such is the organism. This is the essential nutrient growth. These all are the microbial activities, biochemical barriers importance. Okay. Now coming to the post-infective or induced defense mechanism. What is the induced defense mechanism? Here to stop the enzyme growth. This one. What is the post-infectives? Plants have the face the wide variety of pathogens. Plants have to face the wide variety of pathogen. That is the enemies. Pathogen meaning enti? What is the meaning pathogen? Enemies is called the pathogen. These enemies throughout their life, throughout their lifespan, standing at one place, thus nature and the process of evolution have built in them a strategically designed pre-existing meaning the structural and the biochemical reactions of the defense mechanism. This is called the defense mechanism. The real value of this system has not been critically examined. This is not for the critical examined. It appear is that is the pre-existing defense mechanism and help in plants warding off most of microbes as non-pathogens. But it does not seem to sufficient. The disease occurs when pre-existing defense mechanism are not enough to check the entry of the pathogen and the pathogen avoid timely eliminating active defense system in plant tissue or habits active defense response by creating metabolic toxins. The induced active defense mechanism in plants may operate at different levels. One is the induced physical or structural defense. The second one, induced biochemical defense. What is the in that both the activation or induction of defense mechanism may be both specific or non-specific type. Here also two are there. One is the specific type, or the second one is called the non-specific type. Several structural changes are known to be induced by a range of biotic or abiotic illators. These dynamic defense mechanisms prevent further colonization or spread of the pathogen activity. Defense in the plant involves cellular defense that rely upon performed surveillance system which are enclosed by resistance gene. The receptor proteins are strategically located in cell membrane to detect the pathogen or factor translocated by pathogens. The ability of the plant to mount an active defenses response in again under genomic control. Here in this one, what is the physical induced system? In that one, physical or structural histological barriers. In that one, structural barriers. Before I'll explain the lignin, cutin, suberin. Same. Even after the establishment of infection in the plant cell, the host defense system tries tries to create barrier for further colonization of tissues. These may be accomplished at various levels. The first one is the lignification, suberization, abscission layer, the stylosis formation. What is the lignification? In the lignin, this is the secondary metabolites, the cell wall to produce the lignin. After epidermis layer found this lignin layer. Lignified cell provide effective barrier to hyphal penetration. Very, very important. This is the prevent to the hyphal penetration. They also act as a impermeable barriers for free movement of nutrient causing the starvation of pathogen as in potato infection by Phytophthora infestan or cucurbiturm infection by Cladosporium cucumarin. The Cladosporium cucumarin. This is the lignification. Coming to the second one, suberization. In several plants, as soon as the infection occur in the cell, the infected cell becomes surrounded by a suberized wall. Suberized wall. This isolates them from healthy tissue. That is a crocky layer, cork layers. Corky layer formation is a part of natural healing system of the plant, as in common scab of potato and rot of sweet potato. Coming to the formation of abscission layer. Abscission layer are naturally formed in plant as a device for dropping of older leaves and mature fruits. Many plants use this device as a defense mechanism, also by dropping of infected or invaded plant tissue or plant parts. And next plant parts along with the along with the pathogen. Next one is the short hole in the leaves. Short hole in the leaves of a fruit trees are the common in future. Next one is a tylosis formation. The tylosis is the same like that, some pore-like structure. Some tylosis formation. Tylosis is the are namely Tylosis are normally formed in the order xylem vessels of plants by protrusion of the wall of xylem parenchyma cell through the way of the pits. The size and number of tyloses physical block the vessels. Physical block the vessels. The tyloses are induced inductively formed much ahead of infection, thus blocking the spread of pathogen. It suggested biochemical indicators of movement of tylosis. Induced factor. This is called the TIF. TIF upstem as the sweet potato infected by Fusarium axosporium. What is the sweet potato to by that one? Fusarium actosporium. The species batatas. This is the tylosis and that is the formation of tylosis. Next one is the gum deposit. Gum deposits, the gums, these are the secondary metabolites. The gums are gums and vascular gels. The gum and vascular gel quickly accumulated and fill the intracellular spaces or within the cell which surrounded the infection threads and infective hasturias resulting in the death or starvation of infective propagation. See, this one you can observe the all. This is the lignification. The picture you can observe, no? This part is called the lignification. The second one is called the abscission layer formation. The second layer is the abscission layer. This one. This is the abscisic layer. See, here, healthy area, this part is the healthy area. This is the diseased area, lignified cells. How to lignin to enter into the diseased there? Which area? Abscission area. Coming to the next one after abscission, C1, C2, crockery layer. C1, C2, crockery. This one. This is the crocked area. C1, C2, crock area. D, tylosis formation. D1 is tylosis. This one. This is the tylosis formation. These two are the tylosis formation. And E1. E is the steam, see that, seeing that one infection threads. These are the infection threads. These are the picture you can observe. Next one, induced biochemical changes. Induced biochemical induced biochemical changes in the host plant or the last line the host plant or the last line of host defense. This is called host line. This is called the this is responsible various from the plant specific plant plant specific parts or plant tissues from the suspectable resistant variety, their genetic potential. This is a biochemical factors in the host depending upon the some other following attributes also there. The first attribute, the substance is associated with protection against disease at the site where protection occur. The second one, the substance can be isolated from the host showing protection against the disease. Third one, introduction of isolated substance to the appropriate suspectable host corner protection. Next one, the fourth attribute, the nature of protection so induced resemble that of the natural against of a resistance plant. These four are the success defines the different attributes. Biochemical factor host different attributes. In this one, toxic substance produced rapid production, suitable surface, suitable modification or accumulation of the chemical toxic to pathogen upon to effective concentration in is an important component of a overall activity defense in the strategy of the plants. And the next one is the coming to the another one. mm, Strategy plants, so chemical one. The second thing is the role of phenolic compounds. These also phenolic compounds also example chlorogenic acids. తర్వాత next one is the sapphic acids. Oxidation produced in fluorotin, hydroquinones, hydroxyquinones, phytoxins. These are main toxic chemical producers, inhibit the pathogen or its activities. Some of these are P-formed toxic chemical, while other may be denosynthesized or modified to more toxic forms. The enzymes involved in the chemical pathogenic are already present in the pre-existing cell. This is the phenolic compound. Come to the another one, phytoalexins. Phytoalexins is the most common responsible of the stress plant. So, what is the stress? Water stress, temperature stress, air stress, enzyme stress. That is the stress meaning over one type of stress and less one type of stress. Whether biotic meaning pathogen or insects, abiotic wounding. Both are the important. Biotic is the pathogenic biotic or insects. Abiotic wounding is the production and accumulation of substrate that can inhibit the growth and activities of the biotic factor or may help in healing process. The scientists Muller and Borger proposed the concept of phytoalexin in the study of hypersensitive reaction in potatoes are virulent. What is the pathogen? The that is a potato, that is a infestiana. Infestiana phyto antibiotic is the plant pathogen interaction or as a result of response injury their physical simulations. Coming to the next one, role of new protein synthesis. Post-infection changes in host cell infection in the host cell involved production and modification of large number of proteins. Proteins just like the enzymes, structural and function of enzymes, which have important role and defined mechanism. Enzymes required for various synthetic pathways normally are by modified enzymes for producing of relax producing production of a resistant to released substance. In addition, phenolic oxidation enzyme have vital role. The influence of these changes may be confined to infection site or nearby the cells. These increased systems and activity of the phenylamino lyases, PAL. What is the PAL? Phenylamino lyases has been reported in several bacterial and viral pathogen in resistant reaction. These phenylamino lyases play key role in synthesis of the phenols, phytoloxin, and and is the lignins. In effective of resistance depends on the speed and amount of synthesis production, their humid movement of neighboring healthy tissues to create the timely defensive barriers. The second next one in activation of enzyme and toxins. The role played by chemical weapons. What is the chemical? Toxins and enzymes is a chemical of pathogen. During pathogenesis, wall established. The nectotrophs and hemioboiotrophs employ more of these more of these substance by causing more causing more damage in the tissue as compared to specialized obligate parasite. The other hmm, next one is the uh, here, these are the in enzyme toxins coming to the biosynthetic pathway. This biosynthetic pathway, stress physiology. I'll explain no water stress, temperature stress, mineral stress, chilling stress like this. This is a stress when the tissues are changed, stress physiology to cope up the advancing activities of pathogen. Now the new enzyme always enzyme is called the proteins. The new enzymes are produced in an effective to the synthesize defenses released substance. Most of these components are formed through cyclic acid pathway and modification actor pathway. Modification responsible respiration is diseased tissues invariable increased. A part of glycosis is replaced by pentose pathway, which yields a four carbon compound. It is responsible for the earlier stage of infections, influenced the some species. Next one, active and defense pathogen. Induction of pathogen, host resistant, natural or biochemical seems to be universal in plants. Active universal in plants. Active defense response have been reported against all classes of pathogen. All classes meaning fungi or bacteria, viral, mycoplasma, nematodes. These active defense response may lead to the incompletable host pathogen interaction. Next, thus, on entry of the pathogen, a temporary increases in cellular metabolism, in cellular metabolism activities occurs in the host due to the stress caused by the increased metabolisms and increased metabolisms activity cell die rapidly showing hypersensitive reaction. Rapid death of cell is correlated with increased degree of resistance most disease symptom when the infected tissue reaching the nectars also. These are the here phenols, phenolic acids also oxidize in burn. These are the active pathogen. What is the Elicitors. Elicitors is a biology or compound that when introduced into a living organism. Elicitors is a living single the activation or synthesis of another compound. For example, a molecule is jasmonic acid. Okay, where your molecule name jasmonic acid, which stimulate the biosynthesis of delta viniferin in graphene cell culture. Another example is chitosan. which is used in agricultural as a natural biocontrol agent. The recognize of a potential indicators result in the several years responses, including the rapid ion fluxes, activation of kinases, generation of active oxygen species AOS. These early event followed by the defense responses, including induction of the hypersensitive response, that is the HR. HR meaning hyper sensitive response, a localized from programmed cell death. Programmed cell death, PCD, that is the programmed cell death, limiting pathogen spread. Further reinforcement of the cell wall and production of antimicrobial compound such as defense protein and phytolexin. Many of the plant develop responsible or mediated by the interaction set of the single molecular, including jasmonic acid, ethylene salicylic acid. Originally, the term illators was used molecular capable of inducing the production of phytoalexin, but it is not now commonly used for the component stimulating any type of defense. Evenly, the induction of defense response may lead to enhanced resistance. Elicitors differ from hormones. And hormones compound produced in one part of any organism to causing change another part of organism. As elicitor do not have produced within the organism in which they are eliciting a response, and these are usually not naturally occurring in the organism. This border defense of the indicators induces both substance of the pathogen origin and the compounds released from plants by the action of the pathogen. This elicit, this is called the endogenous elicitors. Elicitors do not have any common chemical structure. Wide range different classes compound, including oligosaccharides, proteins, lipids. These all act as a single compound, low concentration, providing information for the plant to trigger defense, distinguishing indicators from toxins which may act only at the higher concentration and affect the plant determination without active plant metabolism. These one most elicitors seen in two border categories. First one, many of them are constitutively present in the pathogen cell wall, structural and compound. For example, glucans, chitins, fragment as well as bacteria, flagellin, lipopolysaccharide. Another class elicitors plays a role as virulence determinants. Examples, hairpins, hairpins product, avergine. While the function of some elicitors remains elusive, the certain plant pathogens interaction, the main elicitors for the plant responsible might be production due to the activity of plant cell wall enzymes. These enzymes that are often essential virulence factor provide the attacking pathogen with nutrient, also release uh, pect- uh, pectic fragments, that is called oligogalactonides, OGA, that can act as endogenous elicitors that vast elicitor compounds are present. These are the types of elicitors. Various general elicitors describe the plant. The oligogalactorinide. This is called the pectic fragments from the plant cell wall. The second, chitosan, chitin fragments from fungus cell wall. You know, the fungal cell wall made up of chitins. Beta heptaglucosan. This is a component of the mycelia cell wall of certain oomycetes. Some oomycetes cell wall made up of beta heptaglucoside or glucosans. This is the mycelia. Next one is the lipopolysaccharides. Lipopolysaccharide gram-negative bacteria. Lipopolysaccharides is called the gram-negative bacteria. Elicitinous cryptogenia proteinous from oomycetes. AVR2, AVR5, AVR4 and 9 products of the corresponding AVR gene. PEP13 oligopeptide of 13 amino acid. FLEG 22 amino acid N-terminal fragment of bacterial plasmin. BCPGI endopolygalactonase from Botrytis cinerea graphinia. AVR-PTO Pseudomonas aeruginosa tomato. These are the, for example, various elicitors general describing. Next one, how to pathogen transduction is there. Pathogen transduction, see, this is the pathogen transduction. How to transductions are there and host plant pathogen. This is the pathogen nucleus. This is the specific elicitors. Pathogenetically genes, this part, active avirulence genes. The pathogen factors, what is the pathogen factor? Enzyme, toxins, growth regulators, polysaccharide. Another factors are the established and maintained basic compatibility. These are the plant defense suspectors. Here, non-specific plant defense are there. This is the host plant nucleus. This is the establish and maintain the basic compatibility interference with the metabolism and translocation of nucleus. This nucleus resistant gene incompatibly specific receptors to come to the triggering of specific deficiency. The host signal chitin, fatty acid, galactophenols. These are the compatibility reaction in between the host and pathogen. That is called the signal transductional pathway. This is called the signal transduction pathway. Here, in this part, plant continuously define themselves against act from the bacteria, fungal, virus, invertebrates, mycoplasma, these are etc. They do not have immunity system, but rather than process performed other inducible defense resistant. The plant disease resistance often depends on the recognition signals, activation into initiate response. The plant pathogen act are perceived through pathogen derived compounds or plant derived molecule that elicit defense reactions. Despite the large variety of elicitors, generally scheme for cellular elicitors, signaling leading to the plant resistance that is known. The early signaling event happen there elicitor, precipitation, changes in the activity of the plasma membrane, protein reversible protein phosphorylation, nitric oxide, active oxygen specific. These events occur within the first minute few hours after elicitor's precipitation. One specific elicitor in transduction pathway can use can use a combination or a partial combination of such event which can differ in intensively depending on the stimulates. That link between the signaling event allow amplification of the signals transduction. This disease spread only in the susceptible plant as the unable to recognize the pathogen responds too slowly. In such case, plant pathogen interaction is generally compatible as the pathogen production characteristic. This is the compatible reaction. See, this one is the another reaction is the incompatible reaction. Compatible reaction is completed. Now, other second one is called the incompatible. See, incompatible, this is the host cell. Okay, this is the pathogen. In the pathogen, these are the elicitors. Elicitors is the pathogen elicitors. Host cell. This is the host cell receptor. Host cell receptor. These are the pathogen elicitors. Now, NBC receptors active site receptor become a active. Elicitors react with the host cell responsibility are receptible. Here, this is the nucleus. In the nucleus, ribosomes. This is the protein binding to DNA, gene expression. This is the localized responsible. This is the oxidative burst. This is the polygramenate cell death. And plant cell, this is the plant cell membrane, plant cell cytoplasm. These are total cytoplasm. This is the incompatible reaction. See, the general view, signal transduction. Signal, auto signal, this is the host cell, this is the pathogen cell. Host cell, these are the receptors. These receptors, this is the pathogen elicitors. This factor, pathogen elicitors, the cell wall to how to move that one. See, receptors signaling to pran, that is the transcription, transcription is the genomic condition, then to mRNA to translation. The total protein synthesis, signaling the protein synthesis, and the defense responsible product cell wall modifier, phytoalexin, PR proteins, HR systematic signals. This is the general viewed. This one is the yeah, compatible reaction and incompatible reaction. This is the signal transduction during the host pathogenic interaction. This is the recognition site, single precipitation, signal transduction, signal responses, termination of signals. This is the most general view of the signal transmission. See, what is the recognition side? This is the first step. In this one, first step of this one, interaction and requisite initiation, the compatibility or incompatibility reactions. It depends upon then if the initial recognition signal received by pathogen favor and growth and development. The reaction is compatible and outcome is a disease. If the signal is suppressed, in that time, the pathogen growth and activity reaction is incompatible and the disease will be ab- uh, ad- aborted. Diseases will be aborted. This recognition depends on the generation of elicitors by the pathogen. Recognition of pathogen triggers a large range of indicible defense mechanism which lead to resistance the plant various mechanism induced at the site of infection like synthesis of antimicrobial compounds is called phytoalexin. Alternation in synthesis of cell wall structural proteins and response in general, which is the invalid. See, this is the first one. This is a recognition site. Next one, signal site. This is a recognition site. Second one, signaling. Signal precipitation. This is a second level. Signal precipitation, see, this is a flora gene for gene hypothesis. See, this is the pathogen genotype, host genotype. AR resistance, AR disease. AR, this is a disease. 1 is to 3. This is the one issue. It is a surface level phenomena which elicitors from the pathogen are recognized by host receptor can be explained by flora gene. See this flora gene for gene. Flora gene for gene hypothesis consists of a receptor ligand interactions. This one is ear resistance gene, pathogen, virulent AVR gene is there. See, this is the receptor ligand interaction. Receptor ligand interaction. This is the uh, plasma membrane. These are the lipids. These are the ligand primary messengers. These are the receptors ligand binding site, this part. Here, the changing in gene nucleus, the signal transfer via second message. The second message, cellular responsible also present in one. Next one, third one, signal transduction. After the precipitation, the next step is the signal transduction. Precipitation of environmental signals medicated by specific receptor likely initiation internal signal transduction pathway. The signal transduction pathway refers to a series of sequential events such as protein phosphorylation constant upon binding of ligand by transmembrane receptors signal through a series intermediate molecules. These are two major pathways, that is the via G protein and the second one is the via mitogene activity protein MAP, MAPK Cas. First one is the via G protein. G proteins, G proteins are composed of three substances. G proteins are composed of three substances, alpha, beta, gamma, where specificity is mainly determined by alpha. The alpha substance constitutes of two domains. GTPase domain is the alpha helical domain. Activation result in this one GTP domain. Via mitosen activated protein kinase uh, cas cascades. MAPK cascades fulfill essential functions in transduction of extracellular signals to cellular and nuclear response. This MAPK is activated by dual phosphorylation of a theronium catalyzed by the PAR kinase activated by serine thiorin phosphorylated All MAPK pathways operate through sequential phosphorylation even to the transcription. See, last one is the signal response through the secondary messages. Secondary messenger systems exist in the plant to plant to the transmit the primary elicitation signals. That is a pathogen activity. Uh, that one is a active oxygen species. Second one, calcium ion influence. Third one, protein phosphorylation, CAMP pathway, salicylic acid, methyl jasmonate, jasmonic acid, ethylene, nitric oxide. C, secondary messenger activation against pathogen elicited response. See, this is the pathogen how to rice cell. This is the rice cell receptors. Cell death expression of the PR protein production of the pathogen. The pathogens elicitors chain, that is the oligomers, beta glucans, oligomers, etc. This is the receptor. This is a phospholipid. Phospholipid to ROS jasmonic acid signal responsible. This is the active. First one is the active oxygen species. What is the active oxygen species? Several active oxygen species involved in signal transductions are H2O2, superoxidized anions. These signalate oxygen and hydroxyl radical or active oxygen species may directly attack the pathogenic organism, and they may indirectly deter subsequent invasion by the pathogenic organism by causing rapid cross-linking of the phenolic compounds of cell wall. This H2O2 is the most. Second one, calcium ion influence. Calcium is a very important second messenger regulatory function. That is the calcium ion are the extracted mostly by the small cytoplasmic protein calmodium on binding calcium. This calmodium undergoes a major confor- conformational changes allowed to the bind the calmodial depend enzyme, calmodial activity protein, that is the CDPK. Calmodial depend protein kinase CA plus 2 concentration switch on the active form of the calmodial cycle. Next one, CAMP. CAMP is a transmission of the CAMP signal, protein kinases, PKA, CAMP differentiates the conedias are emerging appears. Last one is the salicylic acids, H2O2 are there, R genes are present. Next, last one, methyl jasmonic acid. And methyl jasmonic acid, this is a phenylalanine ammonia lipases are there. And the next one is the ethylene. Ethylene is the plant hormone, gaseous formation is there, volatile plant hormone synthesized from amino acid methionine. Ethylene is synthesized amino acid is the methionine. Ethylene is produced upon wounding or infection by the pathogen, as well as by treatment by elicitors of defense responses. The increased production of ethylene is one of the earliest chemically detectable event in pathogen infected plants or treated plants with elicitors. Nitric oxide. Nitric oxide signaling involves GMP depends upon the pathway. Biosynthesis NO is catalyzed by the nitric oxide synthesis NOS. AOS are also known to work with the nitric oxide in the defense response. This is the signal termination should terminate when it is induced and responded to stop. The terminating events are in the corresponded with the deactivation of pathogen induced signals or host. This is the interaction pathogen 2 is completed the two total the two also completed in the your syllabus, not for the third interaction, only 1 2 interactions are there. Now I will close my PPT. Uh, Today, total your syllabus is completed. Wish you very all the best, students, for your upcoming exam, PG exam, theory exam. Good luck. Thank you, sir.